Welcome back to this quarterly series of BioTechBuilder training videos that we post to YouTube. This one is being posted for a couple of reasons. One is that we have a recent alignment to another credential provider called BACE, B-A-C-E, and some of the BioTechBuilder curriculum should be supplemented with videos like these. This one is about using the Autoclave. It will help prepare your students to be successful on this BACE exam. And then the second reason to share this video is to celebrate the fact that our teacher training materials recently were recognized with a Best of STEM award, which is an educator-judged recognition, and we're very excited about it. So here is a video, and we hope it is helpful. All right, welcome back. Natalie.
Hiroko.
And today we're going to show you how to use a new piece of lab equipment called the Autoclave. Do you want to say what the Autoclave does?
Sure. The Autoclave is a machine that will use high temperature, heat, and pressure to sterilize equipment. And lab, it could be equipment, it could also be media, anything that we need to be sterile before we use them in the lab.
Yes. I've also used the autoclave to get rid of contaminated things, so like to decontaminate things at the end. So the autoclave is like a pressure cooker to remove microbes from the equipment that you're going to use or to kill microbes that you no longer need. So here's how it works, right? Hiroko mentioned temperature and pressure. The magic temperature is?
121 degrees Celsius.
Yes, that's 250 degrees Fahrenheit.
It's very hot.
The magic pressure is?
15 PSI, or pounds square per inch.
Yes, pounds per square inch.
Pounds
Yes.
per square inch.
So 121 is the temperature, 15 is PSI, and the time is usually 15 to 20 minutes. I actually usually use 30.
Yeah.
Yeah, just for safekeeping.
Use,
But
yeah,
yes.
25.
Right. So we're going to show you how to prepare the materials to go into the autoclave next, right? So let's say, for example,
We have let's say we want to get a sterile, nice, clean, empty bottle so you can make some solutions in it. One of the things about these bottles with caps is that you want to make sure the lid is loose, so you can kind of hear maybe it's jiggling. And then there is a piece of tape here, and it looks very white right now, but they're special. It's an autoclave tape, and you will tape this to the top and then tape it to the body of the vessel there. So that's one. Now if it's an open container like this flask, you will take an aluminum foil and you will cover it. Same thing, take an autoclave tape, tape the aluminum foil to the glassware, like so.
yes. So she's done an amazing job of it. I will say I've done it wrong a couple of ways. I have tightened the lid of the bottle too much, and then it can explode in the autoclave and shatter or crack, right? So you always want to, as she said, leave the top loose and jiggly so you can hear it. I have also used tape that looks like this, and it looks like masking tape. This is autoclave tape. It is not the same thing as masking tape, and we will show you what autoclave tape does after it's been autoclaved, but I have made the mistake of using masking tape instead of autoclave tape. Am I the only one that's done that?
I have tightened the loose thing, and then yes, the masking tape.
No.
No?
Okay.
But I think because the core facility only had autoclave tape
near the autoclave.
Near the autoclave,
yes.
for
So
that
don't
reason.
use masking tape, it's autoclave tape, and we'll show you how that's special. Okay, so then when it's ready to go into the autoclave, you will use a secondary container. And do you want to say why you might want to do that?
I don't know.
Oh, I don't know really either, but it could leak,
either.
right? The materials could leak. Like if this is going to get,
Yeah.
if there's a lot of liquid in here, it's going to boil, and it could boil out, and then it would make a mess inside the autoclave if it's not in a secondary container. Or if you have forgotten to loosen the lid and the bottle shatters, you would have to clean out all the broken glass from the autoclave. This way, it's in a secondary container, so you would be able to just pull the thing out and clean up just what's in there. So I always autoclave in a secondary container.
Yes, always secondary container, and now I know the reason why. And you do too.
Mistakes, that's why. Okay.
And for dry equipment, so this is dry, meaning there's no liquid inside. there are two things that you can autoclave, things that have liquids in them, things that are dry like this. The dry ones, they go in a secondary container. If these contain media, what you want to do is make a little bain-marie. If you do any cooking, you put a little bit of water inside, so that the water also warms it up.
I had forgotten that,
yeah.
yes. And you know why? Because the bottles, this is where they crack. Right at the base,
Oh,
right at the
where
base.
it's
That's
been,
the weakest place of them. So that's why you put
together.
extra water in the bottom if there's liquids. I forgot that.
Oh.
Yes. Okay. So she's gonna load up these into the microwave, the autoclave.
So this is room temp. There are hot hands. So if your machine is warm, or if you're anyway, probably the most proper way to do it is to wear this regardless. And you take your prepared material, put it in. And this one has this nice door that can close, and this is a touch screen that you can start. You might also come across autoclave machines that have a big wheel that's like on a submarine.
Yes.
But
To seal
yes,
it. Yep.
to seal it up. But
Yep.
there are, yeah.
And then it goes to 121 temperature, 15
15.
psi for at least 20 minutes.
At least
right?
20 minutes
Yep.
is, yeah,
Okay.
recommended.
And then what does it look like at the end if you have other things? How do we know it's done?
How do we know it's done? You know it's done and it was properly autoclaved when the autoclave turns to black stripes like this. That indicates that it reached the correct temperature as well as the pressure, and for the minimum amount of time needed. If this tape doesn't turn black like this, that means that your autoclave failed and that your items that you wanted to sterilize are not sterilized, meaning bacteria, spores, germs are not dead.
Right, so you'll have to do that again.
Do
Right.
it again.
One tip is that we've said that we want to autoclave it for 15, 20, 25 minutes. Do not leave just that amount of time if you're trying to run an experiment and have something sterile, because this will take some time to reach that temperature and that pressure, and it takes time to come back down from temperature and pressure. So you cannot open the door until the pressure is back to atmospheric, so 1, right, PSI.
And that can take
And
some
that can
time.
take some time to vent. So you always, even though if we are autoclaving it at that temperature for a given amount of time, the whole process will take longer than that. Yeah,
Yes.
All right.
Perfect.
and that should be it. I think we're good.
I think we are.
Excellent.